Factors affecting enzyme action. The substrate combines with an enzyme to form enzyme substrate complex. The enzyme brings about changes in the substrate and forms product. So there are three things that you can see in this particular reaction: enzyme, substrate, and product. The concentration of all these three affect the activity of the enzyme. So the factors affecting enzyme action are the concentration of enzyme, the concentration of substrate, and the concentration of product. Along with these three factors, there are other factors affecting enzyme action, such as the temperature of the reaction, the pH of the reaction, activators and coenzyme, and various physical agents. So these are the 7 main factors affecting enzyme action that we are going to study in this particular video. Let's look at each of them one by one. So the first is the concentration of enzyme. The relationship between the concentration of enzyme and the velocity of reaction is very simple. As the concentration of enzyme increases, the velocity of the reaction increases as well. Let's look at this reaction. Enzyme plus substrate gives enzyme substrate complex. As you can see, if the concentration of enzyme is increased, the reaction in forward direction increases. And that is why, as the concentration of enzyme is increased, the velocity of the reaction increases. Let's say initially the concentration of enzyme is 0. At this particular condition, the velocity of the reaction is 0 as well. As you start increasing the concentration of enzyme, the velocity of the reaction increases as well, and that's why we get a graph with a straight line. This concludes that the velocity of reaction is directly proportional to the concentration of enzyme. Let's understand the same relationship with the help of an example. As you can see, there are two vessels, 1 and 2, of same volume. In the first vessel, there are 2 enzyme molecules. In the second vessel, there are 1, 2, 3, 4, 5, 6, 7 enzyme molecules. In the first vessel, the substrate can combine with either this enzyme or this enzyme and form enzyme substrate complex. In the second vessel, the substrate can combine with either this enzyme or 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 this enzyme and form enzyme substrate complex. So for substrate to form enzyme substrate complex, there's a higher probability in second vessel as compared to first vessel. The reason being the concentration of enzyme in second vessel is higher as compared to first vessel. So higher the concentration of enzyme, higher is the probability of substrate forming enzyme substrate complex, and higher is the velocity of reaction. This concludes this particular relationship right here, that the velocity of reaction is directly proportional to the concentration of enzyme. This is the first thing that you need to know, or the first factor affecting enzyme action. The second factor affecting enzyme action is the concentration of the substrate. As the concentration of the substrate increases, the velocity of the reaction increases as well. However, this is true only initially, because later, even upon increasing the concentration of the substrate, the velocity comes out to be a constant. And here's the reason behind it. So let's say initially you have 10 molecules of enzyme, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So you have 10 molecules of enzyme in a vessel. The same vessel contains 2 molecules of substrate. So 2 enzymes are able to form enzyme substrate complex with 2 substrate molecules. As you increase the number of substrate in that vessel, the velocity of the reaction also increases. Let's say at this particular point, at this particular point, half of the enzymes, there are 10, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Out of 10 enzyme molecules, half of the enzymes form enzyme substrate complex because the number of substrate molecules has been increased from 2 to 5. So 1, 2, 3, 4, 5. At this particular condition, the velocity of the reaction is half of the maximum velocity. Let's say we keep on increasing the number of substrate molecules in that given vessel. A condition comes or a point comes at this particular point when all the enzymes get exhausted or all the enzymes finally receive substrate to form enzyme substrate complex. So there are 10 enzyme molecules in the vessel. All of the 10 enzyme molecules get one individual substrate molecule to form enzyme substrate complex. At this particular condition, the velocity of the reaction is maximum. If we further increase the number of substrate molecules, that is if we increase the concentration of substrate beyond this point, if we further increase the number of substrate molecule, the velocity of the reaction is unaffected because all the enzyme molecules have been exhausted or used up. So initially, the velocity of the reaction is directly proportional to the concentration of substrate. However, the important point is that when enzyme gets exhausted, that is all the enzyme molecules get used up, the velocity becomes constant or velocity becomes same even upon increasing the concentration of substrate. Velocity becomes same. So let me revise the whole thing. See, upon increasing the concentration of substrate or upon increasing the number of substrate molecules in a given amount of volume, the velocity of the reaction increases initially. However, there comes a point when the number of substrate molecules become equal to or more than the number of enzyme molecules in the same vessel. At that particular condition, the velocity comes out to be a constant. Here's the reason behind it. Let's say initially there are no substrate, zero concentration of substrate, only enzymes are present in the vessel. There's a vessel, contains 10 molecules of enzyme, 10 molecules of enzyme, 10 molecules of enzyme, and there's no substrate. At that particular condition, the velocity of the reaction is zero as well. Now, we increase the number of substrate molecules from 0 to 2. Then the velocity of the reaction increases because 2 molecules of enzymes are able to form 2 molecules of enzyme substrate complex. Upon further increasing the concentration of substrate, the velocity of the reaction further increases because more enzyme molecules are able to form even more enzyme substrate complex. A point comes where the number of substrate molecules is equal to the half the number of enzyme molecules. At this particular condition, half the number of enzyme molecules, so here are 10, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Out of 10 molecules of enzyme, 5 molecules of enzyme are able to form enzyme substrate complex with the substrate molecule. Now, if we further increase the substrate concentration, the velocity further increases. At this particular point, where half the number of enzyme have been exhausted, the velocity is half the maximum velocity of the reaction. Upon further increasing the substrate concentration, a point comes where all the enzymes get exhausted by the substrate molecules. At that particular condition, the velocity of the reaction is maximum. Upon further increasing the concentration of the substrate, no change arises in the velocity of the reaction cause all the enzymes have been exhausted or used up. So there's a constant called Michaelis-Menten constant or Briggs and Halden's constant, which is represented with the help of K, M. M for Michaelis-Menten and K means constant. So it's a Michaelis-Menten constant. What exactly is Michaelis-Menten constant? So Michaelis-Menten constant is basically the concentration of the substrate during which the velocity of the reaction is half the maximum velocity. So it is the substrate concentration to produce half the maximum velocity. See, in this particular graph, you can see that the half of the maximum velocity is present at this particular point. At that particular point, the concentration of the substrate gives the Michaelis-Menten constant. So if I draw it, so here's where the half of the maximum velocity is present. Corresponding to the velocity, here's the substrate concentration. This particular concentration right here. So the substrate concentration at this particular point gives the Michaelis-Menten constant. So Michaelis-Menten constant is the concentration of the substrate during which the velocity of the reaction is half of the maximum velocity. So the third factor affecting enzyme action is temperature. The activity of the enzyme is maximum at a particular temperature, and we call this the optimum temperature. At optimum temperature, the velocity of the reaction is maximum or the activity of the enzyme is maximum. The graph that we get when we draw a graph between temperature and velocity of the reaction is a bell-shaped graph. So this is a bell-shaped graph. Let's decode this bell-shaped graph. So first, at 0 degree Celsius, there is no enzyme activity. There is no enzyme activity. Upon increasing the temperature, the activity of the enzyme decreases. So enzyme activity increases. Here's the reason behind it, or mechanism behind it. Mechanism. When you increase the temperature, the kinetic energy of the molecules, that is enzyme and substrate, increases. As a result, the collision between enzyme and substrate also increases. As a result, the formation of enzyme substrate complex also increases. Then comes a temperature. Then comes a temperature T0, which is the optimum temperature when the enzyme activity is maximum. So there is maximum enzyme activity. However, upon increasing the temperature beyond the optimum temperature, the enzyme activity actually decreases. And here's the mechanism or reason behind it. Mechanism. When you increase the temperature beyond optimum temperature, the denaturation of protein occurs. Denaturation of protein occurs. And due to the denaturation of the protein, the activity of the enzyme decreases. Enzyme activity decreases. Let me revise the whole thing. So initially at 0 degree Celsius, the velocity of the reaction is 0 because there is no enzyme activity at all. Upon increasing the temperature, the kinetic energy of the molecules, that is enzyme and substrate, increases. As a result, collision increases. As a result, enzyme substrate complex formation increases and the velocity of the reaction increases or enzyme activity increases. Till when? Till comes a temperature at which the activity of the enzyme is maximum. At this particular temperature, the activity of the enzyme is maximum and we call this the optimum temperature. Upon further increasing the temperature beyond optimum temperature, the activity of the enzyme decreases due to the denaturation of the protein of the enzyme. In human body, The optimum temperature for the enzymes inside human body is 37 degrees Celsius for body enzymes, which is actually the normal body temperature of human. Okay, again, see, here's the thing. Initially, the temperature is 0 degrees Celsius. At 0 degrees Celsius, there is no enzyme activity, so the velocity is 0. When you increase the temperature, the enzyme activity increases, so the velocity increases. The reason being, when you increase the temperature, the kinetic energy of the enzyme and substrate molecules increases. As a result, the collision between enzyme and substrate molecules increases. As a result, the tendency of formation of enzyme substrate complex increases. As a result, velocity increases. Till which point? Till this point right here, when optimum temperature is achieved. At optimum temperature, enzyme activity is maximum. At maximum enzyme activity, the velocity of the reaction is also maximum. Upon further increasing the temperature beyond optimum temperature, the enzyme activity decreases. The reason being, the protein forming the enzyme gets denatured, and as a result, enzyme activity decreases and the velocity of the reaction decreases as well. The optimum temperature for enzymes present in human body is 37 degrees Celsius, which is the normal human body temperature. There's one more term that you need to know, which is Q10 value. Q10 value. This Q10 value is called temperature coefficient. Temperature coefficient. Basically, Q10 or temperature coefficient states that the velocity of the reaction increases by times 2 for every 10 degrees Celsius rise in temperature. That is, if you increase the temperature by 10 degrees Celsius, the velocity of the reaction increases by times 2. For every 10 degrees Celsius rise in temperature, the velocity of the reaction increases by 2 times. That's what is stated by Q10 or temperature coefficient. The fourth factor affecting enzyme activity is pH of the chemical reaction. The same concept as that applied in temperature is applied here as well. That is, the velocity of the reaction is maximum or the activity of the enzyme is maximum at optimum pH. Below optimum pH or above optimum pH, the velocity of the reaction decreases due to the decrease in activity of the enzyme. For example, the optimum pH for pepsin, enzyme pepsin, is 1.2. At pH 1.2, the activity of pepsin is highest. At a pH lower than 1.2 or higher than 1.2, the activity of the pepsin enzyme decreases. Similarly, you have trypsin. For trypsin, the optimum pH is 8. At 8 pH, the activity of the trypsin is highest or maximum. Beyond 8 or below 8, the activity decreases. The fifth factor affecting enzyme activity is concentration of product. As the concentration of product increases or accumulation of product takes place, it inhibits the chemical reaction or it inhibits the activity of the enzyme. Accumulation of product leads to inhibition of enzyme. At number 6, we have activators and coenzymes. Coenzymes is going to have a video of its own. But what about activators? Activators are those metallic ions that are responsible for the activation of enzyme. Some enzymes are unable to act on their own. They require activators to activate them. And these activators are present in the form of metallic ions, such as chloride ion, magnesium ion, calcium ion. At number 7, we have inhibitors. Inhibitors, as the name suggests, are responsible for the inhibition of inhibition of enzyme activity. That is, inhibitors, they decrease the activity of the enzyme. And at number 8, Even though I said there are going to be 7 factors, inhibitors is something that I included later on. Okay, so number 8 are the physical agents such as light. For example, salivary amylase. The activity of the salivary amylase increases under red and blue light. However, the activity of the salivary amylase enzyme decreases under UV light. And these are the 8 factors. Even though I said 7, let's add one more. 8 factors that affect the activity of the enzyme. Let's look at all of them one by one in very short. So, number 1 factor is enzyme. The velocity of the reaction is directly proportional to the concentration of the enzyme. As the concentration of the enzyme increases, the velocity of the reaction increases as well. As you can see in this particular reaction, enzyme plus substrate gives enzyme substrate complex. As the concentration of the enzyme increases, the reaction in forward direction increases, so the velocity of the reaction increases. When the number of enzyme molecules is higher, substrate has greater probability to combine with any one of them to form enzyme substrate complex. Second is substrate concentration. Initially, with the increase in substrate concentration, the velocity of the reaction increases as well. However, a point comes when the velocity of the reaction becomes constant even upon increasing the concentration of the substrate. Initially, when the substrate concentration or the number of substrate molecules is less than the number of enzyme molecules, the enzymes combine with substrate to form enzyme substrate complex, and as the number of substrate molecules increases, more enzyme substrate complex are made and the velocity becomes higher as well. However, a point comes where all the enzymes get exhausted by combining with substrate molecule, forming enzyme substrate complex, and upon further increasing the number of substrate molecules, no change in velocity takes place. At this particular point, the velocity is maximum. The concentration of the substrate when the velocity is half the maximum velocity is called Michaelis-Menten constant or Briggs and Haldane constant. At number 3, we have temperature. The activity of the enzyme is highest at optimum temperature. Above and below that temperature, the activity decreases. At 0 degrees Celsius, there is no enzyme activity. Upon increasing the temperature, enzyme activity increases due to higher collision. A temperature comes where the enzyme activity is maximum and we call it the optimum temperature. Upon increasing the temperature beyond optimum temperature, the enzyme activity decreases. Due to the increase in temperature, there is denaturation of protein, and that's why enzyme activity decreases beyond the optimum temperature. For body enzymes, the optimum temperature is 37 degrees Celsius. Q10 is the temperature coefficient, which states that upon increasing the temperature by 10 degrees Celsius, the velocity of the reaction also increases by times 2. At number 4, we have pH. The same concept is applied here. That is, the enzyme activity is maximum at optimum pH. Below optimum pH or above optimum pH, the activity of the enzyme decreases. For example, for pepsin, the optimum pH is 1.2 and for trypsin, it's 8. At number 5, we have concentration of product. Due to the accumulation of product, that is increasing concentration of the product, inhibition of the reaction takes place due to the inhibition of the enzyme. At number 6, we have activators and coenzymes. Activators are the metallic ions such as chloride, magnesium, and calcium that activate the activity of the enzyme. At number 7, we have inhibitors. Inhibitors inhibit the activity of the enzyme and decreases enzyme activity. At number 8, we have physical agents such as light. Salivary amylase activity increases under red and blue light. Its activity decreases under UV light.